Hello everyone. In this class, we'll try to learn about haemocytometer or haemocytometry. Haemocytometer is a device used for counting the hematological cells, and haemocytometry is a procedure. Normally, haemocytometer is in physiology lab used for counting RBCs, WBCs and eosinophils. The device used for determining number of cells per unit volume of suspension is called counting chamber. A haemocytometer consists of thick glass microscope slide with grid of perpendicular lines etched in the middle. The grid has specified dimensions so that the area covered by these lines are known, which makes it possible to count the number of cells in a specific volume of a solution. The principle of haemocytometry is to count the number of cells in a specific volume of a diluted solution and convert it into number of cells in 1 cubic millimeter of undiluted solution. Description of Newber's chamber. It is a single heavy glass slide counting chamber. There is a central depressed platform separated on each side from remaining platform by shallow trenches. The surface level of this platform is 1 by 10 millimeters lower, on which the cover slips rests. The depressed platform is divided into two parts by short transverse gutters, which are called as trenches. Each platform has got improved Newber's ruling. So this is how the Newber's chamber looks like. So there is a H-shaped groove, and in between you have depressed central platforms on which the Newber ruled areas are etched. So this is the top view of Newber's chamber with the H-shaped groove and the central platform, which are little depressed, and you can see the etched Newber's ruled areas. And if you see from the side waves with a cover slip top up on, you can see the depth that is from the lower surface of the cover slip to the etched areas, and which measures around 1 by 10 millimeter. This is how the Newber's ruled area is seen under the microscope. You can see the Newber's ruled area, so which is a square in shape and which measures around 3 into 3 cubic millimeters in length and breadth. So this area divided into 9 equal parts, and there are peripheral corner squares. So these are called as corner squares, and you have central or middle square. The corner square, they are utilized for the counting of WBCs, and the central square is utilized for the counting of RBC, which is usually done in physiology lab. There are 9 squares whose length and breadth is 1 millimeter each, so the total area is 9 millimeter square. The 4 corner squares, one at the upper and left known as upper left square, and one at lower and left is known as left lower square. Similarly, there are upper and lower right squares. These 4 corner squares are marked by extra line on their periphery for easy demarcation, and every corner square is divided into 16 small squares by 3 horizontal and 3 vertical lines. So this is the Newber etched area, which is divided into 9 equal squares, and peripheral squares are called as corner squares, which are denoted as upper left, upper right, lower left and lower right, and each corner square has been divided further into 16 smaller squares. And the central square, which is usually utilized for the counting of RBC, has been divided into 25 small squares by triple lines. So in the physiology laboratory, usually the WBCs and RBCs are counted and the peripheral squares, they are utilized for counting of RBC and WBCs, and the central square is utilized for counting of RBC, particularly the corner squares of RBC counting areas, that is the medium square and the central square of the medium square, they are utilized for counting RBCs. If we calculate the volume in which the WBCs are counted, each corner square measures around 1 millimeter square, and the volume becomes 1 by 10 cubic millimeter. So 1 volume square measures 1 by 10 cubic millimeters. So we are going to count WBCs in such 4 squares. So the volume becomes 2 by 4 millimeter cube. According to the Haemocytometry principle, if 2.5 cubic millimeter contains n number of WBCs, then 1 cubic millimeter contains 5 by 2 into n number of WBCs. Here we have diluted the blood by 20 times, so multiplying that dilution factor gives the number of WBCs contained in 1 cubic millimeter will be 1 cubic millimeter of blood will be 50 into n number of cells. The central square, which is utilized for counting of RBC, it is divided into 25 small squares, and they are marked by triple lines for easy identification of one small square from each other, and each small square again divided into 16 smallest squares by single lines, and hence the length and breadth of each intermediate square is 1 by 5 millimeter each. So this is the central square which is used for the counting of RBC. As we discussed, it is further divided into 25 squares, and amongst 25 squares the peripheral corner squares and the central square, that is 5 intermediate squares, they are utilized for counting the RBCs. So these are the 5 squares which are utilized for counting the RBCs. And if you see the dimension of each intermediate square, it is 1 by 5 into 1 by 5, and if you consider volume, the total volume is 1 by 250 millimeter cube. The area of one intermediate square is 1 by 5 into 1 by 5, that is 1 by 25 square meter, and we are counting RBCs in 5 medium squares, so that makes the volume up to 1 by 5 square millimeter. Hence the volume becomes 1 by 5 into 1 by 10 is equal to 1 by 50 cubic millimeter. And according to the Haemocytometry principle, if 1 by 50 cubic millimeter of diluted blood contains n number cell, therefore 1 cubic millimeter of diluted blood contains 50 into n number of cells. And here for WBC counting, we are diluting the blood by 200 times, so 1 cubic millimeter of undiluted contains 50 into 200 into n, that is 10,000 into n number of cells. So even it is possible to calculate the smallest RBC square also, the length and breadth of which are 1 by 20 millimeters and the depth is 1 by 10. So the volume becomes 1 by 4,000 cubic millimeter. And this is how the WBC counting chamber is going to be seen under low power. You'll be able to see all the 16 squares of each corner square, and you can easily count the WBCs in them. And this is how the RBCs are seen. So when you focus under high power, so one of the intermediate square is seen, which is surrounded by triple lines, and we have 16 small squares in them with RBCs. Note that you can see only one of the 9 squares present in the Newber ruling under low power. Under high power, you can see one of the 16 small squares uh, in RBC counting square and one of the 25 small squares in RBC counting square. These are the WBC and RBC counting pipettes. So this is RBC pipette and this is WBC pipette. Here the blood is diluted appropriately, and in with RBC you are going to take 0.5 part of blood which is going to be mixed with 101 parts of the diluting fluid. So since the blood or the fluid remaining in the stem is not going to take part in the dilution, so 0.5 part is going to mix with 100 parts of the diluting fluids. Hence dilution here is 1 in 200. Similarly for WBC, for counting WBC, 0.5 mark blood is taken and it is mixed with 11 parts. So after discarding the one part, so 0.5 parts of blood is going to be diluted in 10 parts, so dilution becomes here 1 in 20. So this is how the WBC and RBC pipette they look in a haemocytometer. These are some of the difference between RBC pipette and WBC pipette. Calibration in the RBC pipette are 0.51 below the bulb and 101 above the bulb, whereas calibration in WBC pipette are 0.51 below the bulb and 11 above it. RBC pipette bulb is large, the bulb is small in case of WBC pipette as we require lesser dilution. The volume of bulb is 100 times the volume of the stem in RBC pipette, whereas the volume of bulb is 10 times the volume of the stem. Normally the dilution is done 1 in 200 in case while you are counting RBCs, and the dilution is usually 1 in 20 when you are counting WBC. So this is in brief about haemocytometry. Thank you.